Good evening, AP Biology students. Now we're going to talk about transcription, which is the process of making a copy of your DNA, but not replication. No, 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 no. We're making an RNA copy of your DNA. All right, so one of the things we talked about in an earlier presentation is that protein synthesis, the creation of proteins, is basically a process where we generate polypeptides, which is the polymer form of amino acids, and enzymes. And polypeptides are important because they tend to be structural components of all aspects of a living thing. And enzymes are important because they're the biological catalysts that allow chemical reactions to happen fast enough in living things. Now, the first step in this process is to copy your DNA. I mentioned earlier that your DNA really can't be messed with. Like, you can't just directly use your DNA to make proteins. Your DNA has to be preserved because the whole purpose of all living things is to survive so that they may reproduce, because your genes must be passed from generation to generation. Well, your genes are passed in your DNA. So if your DNA isn't stable, if it isn't preserved, if it isn't a solid structure that doesn't have damage or doesn't break down, then you can't pass on your genes. So one of the ways that we protect our DNA is to make sure that we don't directly use it in the process of protein synthesis. Instead, we indirectly use it. And that process of indirectly using it starts with transcription. You have to think about DNA as not like a tool, but more like a printing press. Your DNA is used as a template to make stuff over and over and over. But that stuff that it makes, which happens to be messenger RNA or primary or pre-mRNA, primary transcripts, that stuff that it makes, that's what we use to make the proteins, to make our polypeptides, to make our enzymes. So it's not the DNA, it's the DNA indirectly. So the DNA is our template for our messenger RNA. Well, the process of accessing that template, the process of using that template, is known as transcription. All right? So transcription is the process that we're talking about in this presentation, and it is the process of using DNA to create complementary RNA strands. So that's the beauty of DNA. Right here you've got your, let's do this. Right here, you've got your DNA double helix. Right here, it's actually a double helix, okay? And you know that the double helix is two anti-parallel complementary base pairs. So if you have one side of the helix, you know what the other one is. Well, using that fact that if you have half of the helix, you can always make the other half, the process of transcription takes one half of the DNA and actually creates a single-stranded piece of messenger RNA. And that single-stranded piece will be modified and then used to generate polypeptides and enzymes. Now, to make this process happen, you have to sort of signal that, hey, I need to transcribe this part of your DNA. So for that to happen, we have proteins, other chemicals, substances that are known as transcription factors. And those transcription factors bind to your DNA. And when they bind to your DNA, they attract this enzyme known as RNA polymerase. All right? Again, I cannot emphasize this enough. DNA polymerase is an enzyme that makes the polymer DNA. RNA polymerase is the enzyme that makes the polymer RNA. Transcription is done by RNA polymerase. Replication is done by DNA polymerase. Now, these transcription factors bind to the section of DNA that basically says, hey, start here. Come here, RNA polymerase. So all these factors bind to that section. We call that section a promoter. Now, these transcription factors can be all kinds of things. There are elements that are required to make this happen. There are protein complexes that are required, sometimes hormone signals from your adrenal glands, from your pituitary gland. They get to your cells and they start this process. It's just important that you understand that it's a multitude of substances. Now, once the RNA polymerase binds to the promoter region, it'll actually open up the helix, which is actually what you see right here. So in this instance, it's actually RNA polymerase itself that creates this opening in the helix. All right? Now, once the helix is open, you still can't start. There's a couple other transcription factors that must bind to the RNA polymerase. Now, this second stage of transcription factors tends to be more stuff that's located in the cell. Outside things can start the process, but it's inside things that really get it going, substances that are a normal, functional part of the cell. All right. Now, when all these transcription factors bind with the RNA polymerase to this open strand, we call that an initiation complex. Now, what happens is RNA polymerase will start to move. All right. It'll move down the strand, and as it moves down the strand, it continues to unwind the DNA and continues to build a messenger RNA sequence. All right. When it builds this, what it's doing is it's taking the DNA template strand, which is this one right here, and it's actually adding complementary bases to the RNA strand, which is right here, at the 3' end. So it's elongating the 3' portion of the messenger RNA, which means that this is 5'. Okay? So as the RNA polymerase moves this way, it's adding bases to the 3' end of the new strand. Now, once RNA polymerase finishes coding, it's going to hit a portion of the DNA that basically tells it to stop, to get off. And we call that portion the terminator. It was recently played in a movie by Arnold Schwarzenegger. All right, maybe not so recently. Now, transcription happens all the time, constantly. If you have a long DNA strand, you could have transcription happening in 4, 5, 6 places all at once. That's actually how an organism generates enough protein, enough enzyme to actually function, is because RNA polymerase can be acting at multiple sites on an enzyme, on a DNA strand multiple times. All right, I guess that's it. This one's a short one. Enjoy.